in place of tools and tech. There's a sign-up sheet going around. Please sign up if you have not already done so. For those of you who don't know me, I realized last week I probably should have introduced myself. I'm Marcy Brandenburg. I'm the bioinformationist at the Taubman Health Sciences Library, and also for DCMB. I'm soon going to be starting to schedule for next semester, so if you're interested in giving a presentation, please let me know. And with that, I'll turn it over to our speaker today, who is Brandon Govindarajoo.
Close enough.
Close enough. Who's going to talk to us about?
Thank you. Hi, everyone. My name's Brandon Govindarajoo. I'm a fifth-year graduate student in Young Zung's lab. I think that's how you say it. He never corrects me. And our lab is primarily interested in protein structure prediction. And just a quick history of protein structure prediction. So initially the idea was you would do purely physics-based methods. You would take all the atoms, do electrostatics, van der Waals, model all the atoms, and it would fold. But the problem is we can't run simulations long enough to do that. They're just too computationally expensive. So there's been this trend to finding approximations to the actual energy functions and see if we can find something that's close enough to the actual physical function to fold the proteins. And so here is some examples of my shining rays of hope of possibly graduating one day. So the red and blue are some models generated by the TACOS pipeline, and these kind of faded out green and yellow structures are the actual native protein complexes. So my pixelated picture here is a simple image of a protein-protein interaction network, and what we're interested in is combining information from the protein data bank to help improve our predictions of protein-protein interactions, and where possible generating medium to high resolution models of how these two proteins interact. So kind of a quick note, I'm sure you guys have seen slides like this before, the growing information of every database that we have access to. But so for protein-protein interactions, there's just been kind of this trend going up of the number of unique interfaces that we have access to model protein-protein interactions.
Would you likely explain that why there's a peak on 2005?
Why does it go up and then go down?
Yeah.
I don't know. So just a quick note on this whole idea of homology modeling is if we have two sequences and they look similar, we believe that the structures are also similar, and we can use the structure from the PDB to help guide the folding of our unknown protein. So Young has developed this pipeline called iTaser. It is currently the best protein folding pipeline in the world. And so just a quick run through it, and I'll go through these pieces in detail. It starts with the sequences. It searches through a monomeric library to identify proteins that it believes are similar in structure to our unknown proteins. And then we combine the restraints and we run thousands of different simulations to try to fold the protein. And then we, after all the simulations are done, we try to group them together to see if the simulations converge somewhere. And we'll take that converged result as our first set of answers, and then we'll allow it to go through another round of simulation where we try to further relax the structure into a more native-like structure. And then we'll include another set of higher resolution energy terms, and then they'll output our final model. So this is just a slide. If you've ever seen Young's talk, you'll see one of these where he points out that he does very well. And so Cas7, Cas8, Cas9, I think they're up to 10 or 11, is the worldwide competition of protein structure prediction, where they do a blind study and experimentalists provide structures that haven't been deposited into the PDB, and these different labs try their best to predict what they look like, and Young does well. So what my major thesis project has been is working on extending this iTaser pipeline to handle quaternary structure prediction. So the overview of the rest of this talk is I'll start with a slide of a set of scores you're going to see a lot for comparing our model structures to the actual native structure. And then I'll introduce you to some of the searching algorithms we have, Cost and Spring, that allow us to quickly search the protein data bank to find homologous sequences. And a point to note, we throw away.
All right.
No worries.
Just saying,
Oh, no. You can do that when you're important. So the Cost and Spring, when we find our results, we throw all high homology templates away. So we don't include them in our pipeline for benchmarking. So if we identify a template that has greater than or equal to 30% sequence identity, we don't include it in the rest of our modeling for the rest of our results. So then I'll show you some comparisons with a physics-based docking approach, and then I'll go into further detail about the actual simulation protocols for the method, and then a comparison of Tacos and Modeler, where Modeler pretty much starts with the template and holds the structure as close to the template as possible and fills in the gaps, and I'll go through that. So this is very simple. The three scores you'll see me refer to a lot. So the first one is the interface contact count. So if this is a native structure, I just look at pairwise contacts at the interface, and I see how many of them are in my predicted model. And I divide it by the total that are actually in here. The second one is this global structural similarity source. So we have two proteins here, green and purple, and you do a structural alignment, so you superimpose them as you see here, and I do a one-to-one distance comparison between the model and the structure to get a overall similarity score. And this same idea here, but with protein complexes, sometimes you can have 100% sequence identity, but different orientations. So if you were to align this, so green's over here and blue's over here, and get a TM score of 0.5, which is significant. But for us, it actually doesn't have the information we're interested in, which is the interface contact. So after we do the alignment, we calculate the TM score of each protein chain individually. And so this would be 1, and then this would be 0 here because there's no overlap between the two. And then we pick the lowest of the two to represent this value here. And so all three of these scores range from 0 to 1, with 0 being terrible, 1 being a perfect match. So a quick run through of our two programs for finding protein dimer templates. So the co-threading starts with two sequences from chain A and chain B of the structure we're interested in, and it combines them in both ways, and it searches them through the dimer library as an artificial monomeric sequence. So in the dimer library, we've just connected all the chains, and we compare them to one another. And so the things we look at when we have two residues aligned is if their evolutionary profiles match. So we get multiple sequence alignments for both of our queries using SciBlast, and we have these pre-generated for our library. We see if there's a match in the alignments. We look and see if our secondary structure prediction for our sequences match those that are in the template, and so on and so forth for all these other features. We see if our predictions for the interface and the solvent accessibility and phi and psi angles match the aligned residue when we're doing our sequence alignment. So a quick note on the next program, which is our Spring library. So it consists of two libraries. So with this first one, it's just each one of these is just a single chain protein, and they each have a unique name. And so I go to the PDB and I download all dimers, and so they have no name here right now. And so what I do is I'll take A and I'll compare it to all these, and then B and C and so on and so forth. Or actually, I'll do it this way. So I'll take this one and I'll compare it to all monomers in my monomer library, and I'll name it after the monomer that is most similar to it. So this one is most similar to A, so I'll name it A1, and this one's most similar to Z3, so it gets a Z name. And so there's this naming convention which shows up next. So when we search for protein complexes using Spring, what we do is we first take the target sequence of our first chain and we search it through our monomer library. And so I see that I get this A and F. So I look in the first chain here, I see, do I have any A's or F's? And if so, I allow them to pass through. And so we have our A's and F's here, and their accompanying binding partner. So here I have my second sequence, and I thread it through the monomer library, and I see I have E and D. And I search and see if any of these are also E and D, and then I get my remaining structures that I'll use for higher level evaluations using my energy scores and so on and so forth. So here is just a backed out overview of the pipeline where we do the monomer searching and the filtering here. And then after we have the remaining structures, we look at structural similarity to these two structures, and we have a couple energy scores to compare to see how well the interface is to what appears in the PDB. So right here is a side-by-side comparison of Koth and Spring, with these being interface contacts going from 0 to 1. And so this is Koth and Spring both return a list ranked from 1 to n of templates it believes to be similar to our target complex. And so what's really important here is you see that Koth and Spring identify templates in one where the other doesn't pick those up. So the first thing we did was seeing if we could, I'm sorry, this is rank 1, then top 5, top 10. So I make a cutoff, I only pick the top 10 from both of them. I do our comparison here. But I want to see if I could combine them and still, within top 10 selections, keep all the right templates and have a better overall selection. So what you're seeing here is, so we have Koth and Spring now on the y-axis, and our combination-based method on the bottom, where we run both of them and we re-rank the templates from each list. And so here is rank 1, the best in top 5, and the best in top 10. And what you can see is we can select the best models from both of these within the top 10 and cover the original individual searching programs. Okay, so the next part here is we want to see how worthwhile it was to actually go forth and develop a threading program. So there's a very popular program here called ZDoc, and it's for docking protein chains. So given two protein chains, ZDoc will search the whole space, and they'll make a whole bunch of different calculations and try to figure out which interaction is the native interaction. So, I'm sorry. So when we took ZDoc's benchmark here and we looked at Koth, Spring, and the ZDoc comparison, we got a real big difference between the two, where ZDoc picked up about 37% of the proper interfaces, and then ours were around 10 to 12. And so ZDoc has been working on this benchmark set forever. And I think it's pretty known that it's kind of overtrained. And so when we give it a set of homology models, so we had 643 protein complexes where we generated high or medium to high resolution homology models. ZDoc didn't do so well. So here on the x-axis is ZDoc, the y-axis is our combination threading program, and for the most part, the ZDoc misses the majority of the templates, and the ones it does find, usually the the interface structure is stuck in a homology model. So the other thing we looked at was template docking. So we have our template complex, and we build individual monomer models, and we try to superimpose each monomer chain onto the complex template. And so at first glance, it looks really nice, and you have this increase in interface contacts predicted and an improvement in structure similarity when compared to the starting template. But if I meant to remove these, but if you actually look at clashes and disregard those as actual proper contacts, all the improvement pretty much goes away. So most of the superposition, there's complete overlap of the two images, and it's not a very good place to start. So here's an example of, so we took the green one and the blue one, and we made these models separately. And then we had a template that gave us the orientation and of how these two chains should be oriented. And when you superimpose them directly onto that, you generally get a lot of overlapping clashes. And you can try starting from here, but most of our simulations just completely distorted them because the clash signal was so strong. So this next part here is our protein, my protein folding algorithm for protein complexes. And so the overall pipeline for this is very similar to those that are that's in iTaser. I start with the two sequences from each chain, and I search them through our complex libraries using Coffin Spring, and I find my set of templates, and then I run them through our simulation. I'll go through each one of these steps, and then I find the consensus structure, do further refinement, and get our final model. So here's just another step of the pipeline here, really looking just at the simulation where we have our query alignment and our template alignment, and we keep the coordinates of the alignment, the coordinates that align, and we disregard the other ones. And then from the unaligned regions, we connect them by a random walk, and then we begin our simulation from there. So in our template library, or PDB library, we've stripped all the other atoms. So we try to, in order to improve the, allow for the simulation to run at a reasonable space, we do this reduced representation model where we're only keeping the C alpha coordinates. And we try to approximate how the other residues behave by our energy function. And so we have a reduced representation. And then for our initial model, so if we have our query sequence and it aligns to this template and the alignment aligns to this part and then this red part here, according to this, we disregard everything else. So we cut out everything that didn't align, and we're starting from here. And then we build a random loop by a random walk, and this is our starting model for our simulation.
So what kind of force field do you use for this Cochrane simulation?
I'll show.
Oh, okay.
So the so there's
But go back to the one. So the alignment you're showing, is that just
some images.
because right now it's got lots of gaps
Yeah.
in it, and I would think that would be something hard to deal with.
Right. This is not related to this. My yeah.
Okay, I would just suggest putting it
Put-
in that's a little more related.
really, yeah.
Because I look at that and it's like, how in the world could those things map onto each other
Right. This is,
spatially?
this is my mistake, but yeah.
Okay,
Okay. You're with me. Okay. So the movement. So when we're doing the simulation, we allow each C alpha to make random movements, and we have 7 different movements here. So if this is one of the polypeptide chains, we'll allow for this one to move by itself, or groups of them where we hold 2 fixed and we allow the ones in between to make random movements. And then we also allow for the second chain of the protein model to move around. So we'll allow for random rotations and translations to see if we can find a better interface. So most of the energy functions are very statistical in nature. So for example, here is an image of a protein interface, and we go through our PDB, we take a non-redundant set, and we look at the frequency of different pairs of amino acids coming into contact. And we'll do this with different sets of criteria, and we'll generate several of these types of heat maps that we use to guide the simulation. So the higher, the more often we see this pair in close contact in the PDB. So we try to use that information to fold our model. And then we also take the template restraints from our alignment to further guide the model. So,
Is that heat map a real one?
yes.
It suggests that the highest are between the same residues.
Right. This.
In part, you've got a diagonal that stands out as brighter colors,
Right.
and the oranges are on the diagonal.
I
don't know what amino acids are.
Yeah, I would have to re-look. This was handed, but to me, but I believe most of the ones on the diagonal are hydrophobic, or that are highlighted. I would have to recheck though.
So,
Go ahead.
sorry. Does each monomer flexible or during simulation?
Yeah, everything's fully flexible.
So here is a energy function for the contact, for the interface.
Right.
So what is the energy function for each individual?
Each individual model, it's very similar. So instead of doing this matrix for the interface, they were done for the single chains, and so they're just separate. They're separated. So we have these matrices for the single chains, and then for the interface contacts, and then each monomer has its restraints from searching through the monomer libraries also.
So you all the interaction just a contact. You don't have the bounded interaction like segment structure constraint.
Right. So this is what I was referring to as our template restraints. So this is a plot of the correlation between our energy score and the structural similarity of our protein complexes. And you can see that there's, so for this slide here, this is when we have multiple templates with high confidence. And then this one to the right is where we have one template with high confidence in our template set. And so you can see this kind of downward trend for both, where there's this pretty significant correlation. And so when we have no template, it's kind of just a blah, and you're stuck doing free modeling and hoping your statistical potentials work really, really well. So a real kind of a quick note on how these simulations work is we're starting from our template and our initial model, and we'll allow some moves to happen to try to go further down in our energy field. And sometimes you reach kind of these energy barriers where you can't go over. So we have this method of changing the temperatures during the simulation, and this will allow you to get over barriers and continue down to the native energy state. But sometimes, even with increasing the temperatures, you can't go over. So we have mini simulations starting from different initial conformations to see if maybe there's a easier path that we can go down to fold the protein. So there should be more dots here, but we actually have thousands of protein images that are generated during this simulation. And so after the simulation's done, ideally you would take the lowest energy structure as your final model, but the correlation is not perfect. So what we try to do is kind of borrow from this idea of the Boltzmann distribution, where our lowest energy state is our most common protein conformation from our modeling. So after all the modeling is done, we do all against all structural alignment and structural similarity comparison, and we take cluster centers from each of the clusters as final structures from our simulation. So after we've finished our simulation, we add the side chains and the rest of the atoms back, and we allow it to go through a simple Monte Carlo simulation with a full atom potential. So these are more physical-based potentials, van der Waals, electrostatics, and so on and so forth. And that generates our final model. So there's a program that's fairly famous in the protein folding world. It's Modeler, and it starts from a protein template, and the aligned residues it holds very, very, very tightly, and it'll allow them to move a little bit, but not really. And then it builds the rest of the unaligned regions, and it tries to give you a reasonable final model. So here we see a comparison of Modeler to our starting templates, and so this is the first model generated by Modeler, and the first template with the highest confidence on on this side here. And so you can kind of see that there's a slight improvement in the interface contacts and these two structure similarity scores, and it's a little more so when you pick the top 10 out of both of them. And so here are the averages of the rank 1 and the best and top 10, and so Modeler does a little bit better. And so where I'm at in my life is so for the top ranked ones, so out of the thousands of simulations, my program can usually pick out a very good structure in the top one. So ideally, when you have your list, you're able to pick the best one, and which is the highest rank, and move forward. And that's what you can kind of see from here is when you compare the rank 1 to modelers, is that most of them are above this line here. But there's a general decrease in the when we look at the best overall in the top 10 for both programs. And so one of the ways we try to significantly improve from the starting template is we use consensus template restraints. So we look at all the templates and we try to combine all the information to better fold the protein. But a lot of it's wrong. And so you see we kind of have a hard time distinguishing wrong from right. and this is some issues with my energy potential. But modeler holds on to the starting position very well, so you kind of just see like this little hump over the starting template. So that's kind of where it's at now, and I'm still working on this potential to shift these values up. Quick note, so most of the error is in the interface. It's very dense. It's very easy to generate a clash when you're moving stuff around here. So a lot of my interface moves get rejected. So I'm working on reducing the clash penalty to allow these to go up. But you do see that generally the overall structure similarity is kind of above this line here. And there's quite a few structures where you find something in tacos, but you don't in modeler.
So you've got things that go along the diagonals, things that correlate pretty well.
Right.
And you got the two axes that have things that have no correlation between the two programs. In essence, like one says 0, and the other one's giving scores to it.
You mean like here?
Yeah.
Okay.
So is there any clues from looking at the things that one program gives high score versus the...
So what happens here is, so what's top one here is just the ranking from the template. So modeler doesn't do any re-ranking. So if the template, put the right template there, you get the right template, right modeler score. But tacos, because we do this clustering here, the structures that are closer to native should cluster together. So we're seeing that they are going towards the right spot, and we can usually get the biggest clusters usually have our best model. And so that's what you're kind of seeing in this re-ranking here. But in the top 10, there is usually a template in our set where modeler is able to find a medium to high resolution solution. And so,
excuse me.
Yeah.
So, how long does it take for your program to?
Okay, so modeler takes about 20 minutes, I think, after you've done all your threading and stuff. Whereas a simulation with tacos takes about 10 to 15 hours. So,
Is modeler running on as many machines, or what's the implementation behind here?
so So the modeler is very fast, and it doesn't deviate much from the initial position. So what's happening here is all these scores here you can only improve them. So if you make a mistake, it doesn't count. It only looks at correct information with all three of these scores. So with modeler, I'll go to this initial structure. If this is our native contacts, modeler will hold these for dear life and won't let them move, and it'll just kind of wiggle this stuff around. And so when we're doing our counting, we're only really counting this part, and if it makes a mistake here, we don't acknowledge it. So if we do like an RMSD where we do a all C alpha comparison, there's a big difference between the two, but we're primarily interested in improving our interface, so that's why you see these scores. So kind of in conclusion, so we have these two web servers here, Coffin Spring, and I'll eventually be finishing packaging and these two programs to run locally on computers, and they can provide complementary information. They can also help with giving further evidence if two proteins interact, and we're actually, I think someone mentioned the craphome, so I've given them some data sets to try to combine that with what they already have. And so ZDoc is a really common program here, but its results are very limited at using homology models, and you pretty much won't beat them when they're using their own benchmark set. And so my program here that I'm still working on, the global similarity between our model and the native structure is usually better than the other ones, but the error is really in trying to combine the consensus restraints from the template. So they usually have about 30% accuracy in the templates, and so we have 20 different templates, each with about 30% accuracy. So we're holding restraints for the wrong context quite often. And so I'm working on balancing this and trying some different potentials to see if we can select out which restraints we should hold and which ones we should let go. And so if you're interested, Young has a very extensive set of programs for protein structure prediction and function prediction. And so the two that I've presented that we have up right now are Spring and Cough. They're very, very easy to run. You just paste in your sequence for both of them and press submit. And the same for Cough. And so this is my lab here. This is a picture I found of smiling. And and so these are funding agencies, and I think they give me money too, but it's safer to keep them in. So that's it. Preguntas. 8 years of Spanish, that's all I have.
So did you play around with a bunch of ways to incorporate the information from Cough and Spring? I guess I'm kind of stealing Jeff's question, but I noticed that a lot of times one could do well in the other. I was wondering if there were like functional reasons. You could just do like a decision tree and say, I use Cough here, use Spring here.
Right, so that's all the way at the beginning. That's this slide here, right? So we run Cough and we run Spring, and here's our decision tree here. And so in the top 10, I can usually pick, the best information out of both of them.
But like when you have one you don't know what's right, like how do you know?
So so they each give us confidence scores, and I've re-weighted them, and we re-rank their confidences, and that's this
Okay.
label here. And so what you're saying here is if this is Spring here and this is Cough, we can usually pick out the correct information, so you don't get these bars on the y-axis. It's just on this x-axis. Thank you. We're good?
Any other questions?
Everyone's happy? Not happy? Pizza coma? Very cool. Hey, thanks for letting me present. Appreciate it very much. Yay, clapping.